Hi, I'm Ruben, a PhD student in the lab of Demeulemeester, and in our lab, we're mainly interested in charting tumor or somatic evolution. And actually, in the field of cancer genomics, we're quite fortunate as we've seen a boom out of new sequencing methodologies in the last couple of years, especially on the level of single-cell and multiomics. However, one thing is still missing, and that's an accurate, high-throughput multiomics technique who is able to capture the vast amount of genomic mutations that arises in these tumors. And so what we really want is we want to have one single method that is able to capture any variant anywhere in the genome while still having readout at the other omic layers like our transcriptome or epigenome. And so in the lab, we developed our own method, which we call Splonget, which indeed is a method, high-throughput method, long-read single-cell, able to profile your genome, open chromatin, and transcriptome layer. So how does Splonget work? We actually start with the classic 10x multiome, and we use the power of Oxford Nanopore to tilt it to a new level. And traditionally, in your short or in your normal 10x multiome, what you do, thanks to their unique barcoding system, you generate an RNA and a tag library of the same single cell. But because you have to make it compatible with short-read Illumina sequencing, you only reject your smaller open chromatin fragments. And by doing so, you're losing critical information. We reasoned if we cannot only profile the small fragments, but literally all fragments, also the long fragments, we can gain critical information about the genome layer as well. And just by some minor adaptations to the library prep and actually using the length-independent sequencing of Oxford Nanopore, we're now able to profile whole genome while still retaining your tag fragments, and on top, full-length transcriptome of the same single cell. Our method is highly scalable for up to thousands of cells and can even be performed in a targeted fashion. And I just want to give a quick shout-out to our lab magician Joris, who made this possible. So let us have a look at our new DNA library. On this plot, you can see on the y-axis the number of reads, and on the x-axis the read length. And this plot is definitely cropped as the tail goes beyond even 50 kb. And so we can use all these reads to actually profile our whole genome layer. When we still want to do our classic attack, what we do is we can divide these reads based on read length and only use the reads associated with your mono, dinusomatic, and trinusomatic attack signal. And so maybe the most difficult part about this multiome is actually doing the downstream analysis, as this is definitely not standard. And to do this, this was not my work alone, but with the whole lab actually, especially Marius, Alex, and Luke. And I'm going to say that single-cell long-read transcriptomics is getting more and more common, and there are some pipelines out there to pre-process it in the form of single-cell nanoseq, but long-read, single-cell long-read DNA, that's an all other ballgame and actually breaks all the pipelines. And so we came up with our own pipeline to process this data, single-cell DNA long, which is in NF-core style, so everyone can use this pipeline in their multiome experiments. Currently, we're merging the two NF-core pipelines, so future users can use one pre-processing pipeline for their experiments. Also, all our downstream scripts are available on the lab GitHub. But so let's put our multiome into action. And we're also very fortunate, actually, to collaborate with the neighboring lab of Ruben Cools, who provided us with a case study about a pediatric patient diagnosed with BLL. And this patient was diagnosed in the hospital at time point D0, for which it received standard of care therapy, which resulted in the first relapse at Q1. Then, once again, standard of care therapy was applied, resulting in the second relapse Q2, after which a specialized CAR T-cell therapy was applied, which was unfortunately also not successful, resulting in the third and final relapse at Q3. We profiled with our multiome this patient at diagnosis D0, Q1, first relapse, Q2, second relapse, and after the specialized CAR T-cell therapy. And already from some prior analysis, you can see that this tumor is very subclonal complex. But first, let's have a look at the transcriptome state of this tumor. I have to note that everything done after this processing can be done with your standard ScanPy of SERDAT downstream analysis. And what you can see is the cells associated with their diagnosed time point are color-coded in yellow, cells associated with the first relapse color-coded in blue, second in purple, and after the specialized CAR T therapy in brown. When we annotate for cellular markers, we can see our 4 tumor clusters at the respective time points, and right on the bottom right, you can see a repertoire from human cells, the microenvironment, as you will. Well, we now have the power to not only profile these on the transcriptome level, but also on the open chromatin level. And it's so nice to see when we do the reclustering based on your open chromatin alone, that we find the same clusters with our single-cell transcriptome annotations. So that's nice validation that actually our data is accurate between different modalities. And you can do some fancier or more proper integrations with your data. For this, we use Scenic Plus, which is actually a model which covers gene-specific regulatory programs or networks. And we were therefore very interested we can find, as an example, a cancer-specific regulatory program. And on the IGV plot, you can see that the 4 tumor time points have open chromatin represented by this bulb in the gene ERK, where our normal cells do not have open chromatin there. We find actually that ERK is regulated by a transcription factor TCF4. And it's nice to see when you go to the transcription or look at the description that actually we see that this is truly a cancer-specific regulatory program, as only these cells have actually expression of ERK and TCF4. But okay, let's take a step back. And I told you in the introduction that we applied CAR T-cell therapy, which is actually a therapy against CD19. So we were wondering how and why did these cells become resistant? Can we find differences between the pre-CAR T cells and the post-CAR T cells? And I first would like to draw your attention to the DNA layer. So what's the difference between the pre-CAR T DNA and the post-CAR T DNA? Well, you can see right at the intron-exon junction an SNV popping up. And what's the result of this very specific mutation? If you look at the RNA level at the post-CAR T cells, you can see complete read-through of the entire intron, so intron retention, as the separation between your exon and intron is not very well visible. It's also interesting to see that also in the second exon, we find different reads or different cells that have also a mutation right at this exon-intron junction, once again resulting in intron retention, hinting at the potential mechanism why these cells became resistant to CAR T. But so we have now long-read data, and long-read data enables us to do all sorts of cool things. We were wondering whether we can actually find the CAR T cells in our data. And we know that these CAR T cells are actually characterized by a very specific fusion of these 2 genes in their receptor. With our full-length transcriptome, we were able to pinpoint this fusion and trace it back in the cells right at the right bottom, which are actually the cytotoxic T cells. And we could validate this based on our DNA if we saw that exactly in these cells we have multiple translocations also in the CAR T context, validating that actually these cytotoxic T cells may be the actual CAR T cells in our data. And for the good eye, you can also see that not only these cytotoxic T cells have the CAR T cells at the right bottom are there, but also in our tumor cluster, which is super interesting. And we were wondering that maybe we're capturing the interaction between a CAR T cell and a tumor cell in the form of a doublet. But that's still open for discussion. And then I like it to take a step back and actually call mutations at a new genome layer. And the sparsity of a single cell makes it actually already accessible to call copy number variations. And on the left plot, every row represents a single cell, and it's annotated with the left vertical bar according to their time point. You can see the copy number genome-wide. When it's diploid, it's gray. Gains are represented in yellow, and when there is a loss, you can see a blue. One notable difference, for example, is the cells. The yellow cells of D0 are wild type of chromosome 7, while the cells in the later time point have an isochromosome. They lost the P arm of chromosome 7 and gained the Q arm of chromosome 7, which is very nice to see. On the right, you can see an example of the target version of a multiome, which uses the same library as input. We wanted to specify or look for a very specific mutation, an SNV, in these cells, which we know a priori was only present in the tumor cells and not, of course, in the normal cells. And it's very nice to see that even with the target version, we even have the power now to even short this very specific single nucleotide variant in very specific cells. And a last specific trick, what you also can do is you can take all your tumor cells, you can take your normal cells and aggregate the counts. And what you end up with is a pseudobulk which is now your normal long-read Oxford nanopore data, which you can use to do or call even the most complex variation. And just as a proof of concept, on the right you can see Q3 tumor, where we have plotted the translocations on the inner circle, We now have greater power to validate the copy numbers, which you see in red and blue, once again confirming the isochromosome. And now we have also the power to even call single nucleotide variants, which are plotted on the outer circle. And so I want to end my presentation with a summary. And the summary is we can now combine 10x with the power of Oxford Nanopore to create a new multiome, which is able to capture the genome layer. If you still want to have your text signal, you can simply divide the reads by length, and it's easily workable to just integrate between your two modalities. And if you really want, you can create a pseudobulk to even chart the most complex variation in your data. And I want to end with maybe the most important slide of all, the lab members. And I want especially to thank Jonas, the PI, Luke, Alex, and Marius to participate in this project. Thank you, and I open for questions.